Hello, so today we're going to do some excretion exam questions. So pause the video here, try these questions out, and then continue on to see how I answer them. Right, this question over here, whoever made this deserves a big pay rise because this question is great. And I'll tell you why it's great, because there's so many things you can learn from it, exam technique wise, but also just how to read a biological diagram in a sense. So, as I always say, read the question first and see what you need to do, right? So, using the information in figure 2.2, what can you conclude about the likely effect of sulfium on the reabsorption of sodium ions in the PCT? Okay, that's a bit of a mouthful, but let's, dissect this. Okay, so we have to use figure 2.2. When it says what can you conclude, the way you answer those questions are quite state the obviously, like you just say how it is, okay? I'll tell you what that really means a bit on later on, but it's saying what can you conclude about the effect of that drug on the reabsorption of sodium ions in the PCT. Okay. Our focus is on like sodium ions, so at least we know that. Right, now I would suggest to read the information above, because if you don't read it, then you wouldn't have known that it's a drug. And the main part of this whole question is the fact that this drug inhibits carbonic anhydrase. Now, think to yourself, what is carbonic anhydrase? Other than the fact that it's an enzyme, right, obviously, but when is it used? You can see it in the diagram over here. Now, the reason why I said I love this diagram is because you have to work backwards. You have to see what effect that one change would have on the whole system. So let's do that. Let's say there is less or no carbonic anhydrase over here. That means that there's less/none of H2CO3, right? So that means that won't dissociate into H plus and hydrogen carbonate ions. Do you see what effect that has now? Okay, right, so if there's less, let's say H plus ions, okay, well clearly this is a co-transport protein, so if there's less H plus going out from the PCT cells into the lumen, well then that means that there's less of sodium ions moving in from the lumen into the PCT. Okay, but what effect does that really have? Well, if there's less of that, that means there'll be less active transport of sodium ions going from PCT into the blood. And that's basically your answer. I might have just talked at you, so let's just quickly do a recap. Okay, what you need to do for these questions is see what changes there occurred, okay? The changes over here. All of this is irrelevant because this is before the change. Right, we've identified the change, now we have to look after it. So, no or less, I say no or less because that's always on the mark scheme, right? You can pick which one you want to say, okay? Yeah, so if there's less of H2CO3, that means there's less of H plus ions. If there's less H plus ions, there's less co-transport of H plus ions out into the lumen. Therefore, by default, less sodium ions going in. Okay, if there's less of that, then there's less Na plus ions being actively transported out. And that is the effect of the drug on the reabsorption of sodium ions. And that's why the answer is this. I hope that makes sense. Now, moving on. This is a classic, man. Honestly, this normally comes up, to be fair, on like paper 3 and stuff, but it comes up on paper 1, 2 as well. It's really great. Now, let's read it. State 3 structures within the tubal cells that are not visible in this image. Okay, you might be looking at me thinking, what are you talking about? How am I meant to say what structures are not present? How would I know, right? Justified. This is why you need to look for hints. The hint is in this information over here. This figure is from a light microscope. Now we're linking two topics together right now. You need to think, what do I know about light microscopes? Well, I know that you can't see subcellular structures. So that could be anything like Golgi apparatus, mitochondria, vesicles, ribosomes. The list goes on, right? There's all of those things that you can't see. So just pick any of them. That's really it. Okay, you can't see them clearly. I can't see mitochondria. Can you? Yeah. Right, and then lastly, okay, biological drawings 101. This was kind of hard to do on an iPad, to be fair, but of course, you would do this with a pencil in your exam. And there are a few things that you need to know. Firstly, your drawing must take up at like more than 50% of the page, okay, or the box. So that means I need you to make your diagram like enlarge significantly. So the cell I drew was this one. I don't know, I fancied it. The next thing you need to know is that all your lines must be continuous, right? Do you see there's no, look at the cell membrane, right? There's no part where the lines are like, hold on, let me do this again. Let's pretend like that, okay? This is so illegal. So illegal. I can't even explain to you how bad that is. You cannot do that. You have to use or draw it in a way that's continuous. Okay. Next thing is you need to have correct proportions. So, for example, if you look over here in this cell, clearly the nucleus is quite large. If I was to, instead of doing that, just do a little small nucleus, wrong, you can't do that. You have to do it in proportion to what you see over here. Okay. And next thing, labels. Right. You cannot have. Let's undo all of this because it's a bit messy. But you cannot do this. You cannot have arrows. You can only have straight horizontal lines. Okay. Don't do it like that. Don't do it like this. Horizontal. That's not even horizontal, but you get the point, right? With a ruler. You must do it with a ruler. And labels. Okay, maybe this was very bad of me. You shouldn't have done that. It's hard on my iPad, so we'll move on. But yeah, labels. These are the most common ones that come up, and people forget about cytoplasm a lot because they're like, oh, it's just empty space. Well, it's not really, is it? Just truly think about what a cell has, and those are the labels. You don't need, like, it's not a hard question at all. You just need to think very logically, and just remember, whenever you overthink on questions, the saying that much stupider, not saying that you're stupid, but like stupid people have done this and succeeded. Yeah, they're not thinking about, oh, can I see a mitochondria in that? No, they're calling it a day. Yeah, they're like, whatever I need to pass it minimum, they'll do that. So those labels over here are like the bare minimum because there is nothing else you can see. So yeah. Right, I hope this video was useful. If you have any questions, let me know, and I will see you in the next video. Bye bye.